some claps. So this is going to be some good beginner knowledge for a few people. Cool. I wish that there was more like contrast between some of the emojis, but it's not the best pull the class option, but it works with the technology. Okay, thanks guys. Okay, so today the content of the lecture is going to be starting with a basic intro to bacteria. For those of you that have had extensive knowledge, some of this is going to be extremely familiar to you, but I want to make sure everyone's on the same page. And then I'm going to focus a little bit on some traits of plant colonizing bacteria. Definitely a lot of plant pathogens have these traits, but there'll also be similar traits that plant beneficial bacteria use, or tourist bacteria, commensal bacteria, ones that don't really impact plant health. So bacteria, it's always good to start with bacterial anatomy. Bacteria are single-celled organisms. They have DNA that's outside of a nucleus, so it's wrapped in histone-like proteins, but not histones, just free in the cytoplasm. The different bacteria clades have different structures on their cell envelope. So we classify bacteria as either being gram-negative or gram-positive. And the gram-negative organisms have this inner membrane, and then they have a thin peptidoglycan cell wall, and then an outer membrane composed of lipopolysaccharides, which is this LPS layer. Gram-positives have still that inner membrane, and then they have a much thicker layer of peptidoglycan. And so these cell envelope properties influence the susceptibility of some bacteria to different antibiotics, as well as it allows us to distinguish them by staining. So this technique is still used, but it's a little bit old school for identification. But the gram stain is a stain that can help us differentiate between these two major bacterial clades. Gram-positives, when we stain them, will take up this crystal violet and retain that dye throughout some decolorization steps. And so they'll be purple under the stain, whereas gram-negative bacteria, although they initially will take up that crystal violet, the ethanol in the decolorization step will remove the crystal violet dye from their much thinner peptidoglycan layer. And then we can counterstain them with this pink stain. And so the gram stain can be used to distinguish cells, bacterial cells by their cell envelope as purple or pink cells. A much simpler method is to see how resistant the bacteria are to lysis by basic conditions. So gram-negative bacteria with their lipid membrane, it's really susceptible to lysis in extreme basic conditions. So if you take potassium hydroxide and murder the bacteria in it, gram-negatives will be very goopy, whereas the gram-positives probably still die, but they don't lyse and they don't become as goopy. So that's a quick and dirty test. That this one's a little bit more commonly used in diagnostics if you just want to quickly differentiate between these two major clades. Bacteria have different structures on their cell surface. So they have some protein structures that can help them attach. Fimbriae adhesins, these aren't things that help them move, they're non-locomotive, but they help bacteria attach to different surfaces. Many bacteria have exopolysaccharides. So these are polysaccharides that are outside the cell, and they can provide a lot of protection to the bacteria against environmental stresses, including dry conditions, not necessarily complete desiccation, but they help them in low humidity conditions. They help them resist antimicrobial compounds that the plant immunity or competing bacteria might produce. Many bacteria are able to move. A lot of bacteria, especially bacterial plant pathogens, have flagella. And so these videos, this one actually has the flagella stained with GFP, and you can see the bacteria, the flagella spinning, and some of the bacteria actually moving across the slide. And here we don't see the flagella, but the bacteria are being propelled across the media. That's not the only way that bacteria move. There's also an extremely cool mechanism called the type 4 pili that works kind of like a grappling hook. So we've got this little Batman here showing a grappling hook. It actually extends, this pilus extends from the end of the cell, attaches to a surface, and then retracts. And so it's basically just, yeah, a straight up grappling hook motion. So the flagella and swimming motility can help bacteria swim short distances. In the context of plant pathogens, this is not going to help bacteria move across a field of plants, but it can help the bacteria move across a root or move across a leaf surface in order to find an entry point that can help it invade the plant. And one way that the motility allows bacteria to find the right place to invade plants is through a process called chemotaxis. And many fungi and oomycetes have chemotaxis as well. So chemo stands for chemical and taxis stands for movement. So this is a process that allows cells to move towards chemicals or away from chemicals. And so here you can see that there's a lot of this chemical. It can bind to a receptor and ends up steering the bacteria over time in direction of the chemical. It is a little bit more random than just direct bacterial movement, but on average, you'll see that the bacteria end up moving toward the chemical gradient if they're able to sense it. And the details of this aren't necessary for you to know, but I think it's useful to highlight that there is a lot of random but directed movement and random but directed processes in biology. Biology is a little bit noisy and messy sometimes, but there still are general observations and rules that we can observe. So then there's also the second motility, which is twitching motility. So this is that grappling hook motility. And this is a motility that really helps bacteria move along surfaces because it requires that pilus to attach to something and then to retract. And so you'll see plant pathogens use twitching motility to move much shorter distances because it's very slow. But it still is something that contributes to many bacterial pathogens' virulence. Bacteria can come in a handful of shapes and sizes. They're not as creative with their cell biology as we'll see in the fungi and oomycetes, but there's some variation. So bacteria, many of them are rod shape. There's also some plant pathogens in the Streptomyces genus that are filamentous, and these used to be confused for fungi because of their filamentous structure, but they are bacteria. And then there's also some very weird bacteria in the spiroplasma and phytoplasma groups that are spiral or amorphous because they don't have true cell walls. And bacteria, although they are single-celled organisms, can have what we can call as a pseudo, a kind of multicellularity. So bacteria often, although they're single-celled, they rarely exist as individual cells in the environment. As they grow and divide, they'll create microcolonies that can grow into biofilms that are coated in that extracellular polysaccharide and some other extracellular polymers, including DNA. And these biofilms can really protect bacteria from a lot of environmental stresses, as well as you'll end up getting some differentiation in bacterial behavior. So although they're all maybe all genetically identical in a biofilm, you might have some phenotypic variation. that can lead to some of these cells becoming dispersers and then dispersing from the biofilm to colonize a different part of the plant. And this is, coordinated by a complex cell regulation cycle. Another trait of bacterial behavior that's very important for their ability to grow and colonize plants is that most bacteria on plants undergo a process called quorum sensing. And this is a very cool behavior that allows bacteria to sense their population structure and then have behaviors that are beneficial depending on whether the bacteria is alone or present with many similar bacteria. And the way this works is that the bacteria cells produce somewhat specific secondary metabolites, which are shown as these red little shapes. And when there's a low cell density of cells, so very few cells, you'll have a low concentration of that signaling molecule. And then the bacteria will slowly be growing, dividing, and we start to get accumulation of that signal because every one of those cells is producing and secreting this extracellular signal. When a sufficient concentration of that signal accumulates, then the bacteria sense that concentration of this molecule, and there's a signaling cascade that changes the transcriptional response of the bacteria. So they end up expressing a completely altered cellular program. Quorum sensing in many bacterial pathogens will change 10 to 20% of the total genes. And so quorum sensing tends to be a global regulator of bacterial behavior, where there's certain traits that are beneficial for bacteria to express when they're highly dense, such as when they're colonizing and invading plants, and then other traits that are more beneficial when they might be in their environmental survival mode. And we're not going to go into too many details of those, but it's really useful to know that bacteria behavior can switch and respond depending on the environmental cues that the bacteria are experiencing. So bacteria have haploid genomes. So that means that they have approximately one copy of each gene. Occasionally you might have a small duplication, but for all intents and purposes, they are haploid. Most bacteria have a single circular chromosome that's around 600 million base pairs, or 6 megabases. And there are some fun exceptions. And some of these exceptions are plant pathogens. Bacteria in the Ralstonia genus have 2 circular chromosomes. And Agrobacterium tumefaciens has 2 chromosomes as well, but they're even weirder. One of those chromosomes is circular and the other one is linear. In addition to having a chromosome, many bacteria have accessory plasmids, and these can carry traits that give a lot of ecological strength to the bacteria. So in Agrobacterium, the strains that are pathogenic, those strains carry what is called the tumor-inducing plasmid. And this plasmid encodes a number of genes that we'll talk about in a future lecture. But these genes encode many traits that allow the bacteria to cause disease. Whereas there's also some environmental non-pathogenic Agrobacterium that look almost identical if you look at their genome, but they're missing the tumor-inducing plasmid. And then these are just some nice blown up images of bacterial genomes. And I like this because we often draw bacterial genomes as like a big squiggly with no structure that's all over the bacteria. But you can see that there is some chromosomal structure to this E. coli. So there's a lot of proteins in the middle that condense the bacterial genome. And when they exploded this E. coli cell and visualize its DNA on a transmission electron micrograph, you can see it has this beautiful structure. It's kind of a classic science art picture. Okay, so in addition to being haploid, bacteria also have some unique genetic architecture traits. So relative to many eukaryotes, bacteria have a really high gene density. And so what we mean from that is that there isn't as much of what most of us learn about junk DNA, which is a terrible term. But a lot of organisms like eukaryotes have non-coding DNA. Bacteria mostly have DNA that codes for genes. So that's where we say gene density. This part of the DNA encodes for this gene. There's a very tiny gap, and then another gene, very tiny gap, another gene. Some eukaryotes have lots of bases in between all of those genes that influences the regulation and expression of each gene. But bacteria have much simpler genetic structure. Additionally, bacterial genes are often encoded on shared promoters. So promoters are what helps the RNA polymerase express the DNA into messenger RNA or mRNA. And so we get mRNAs that are polycistronic and encode multiple genes. So we've got the green gene, the red gene, and the purple gene, all in the same mRNA. And then ribosomes for bacteria will jump on at each of these translation start sites and translate those mRNA genes into proteins. And additionally, there is not this process of RNA processing. The mRNA doesn't have introns, and so the ribosome, it goes directly from mRNA to translation. And it's really useful to know that bacterial genes are often co-expressed on single promoters, because when we start looking at bacterial genomes, we find that a lot of genes are clustered by function. And so this makes bacterial genetics fun and exciting, because if you look at one gene, this HRCU, often we can hypothesize that the genes around it are involved in similar processes for the cell. And so we're able to look around the genome and come up with testable hypotheses about what function genes are doing based on their gene neighborhoods. And so one of the reasons they're clustered by function is that many of them will share a promoter. And so bacteria are fairly efficient. They're not perfectly efficient, but there is some efficiency here in using one promoter to express all of the genes you need to make tryptophan at the same time. So bacteria are haploid, but there is some amount of sexual reproduction that can happen. And so they are able to, although they don't ever have a diploid state, they are able to exchange DNA and take up DNA from other bacteria. There's multiple mechanisms for this. And we call all of this horizontal gene transfer, because instead of the genes being transferred vertically from parent to daughter, the genes are being transferred between two cells horizontally. And so conjugation is one of the mechanisms. This happens by a specialized structure that gets produced that attaches to cells and actually secretes the DNA through that pilus structure. And that process is fairly rare, because overall, very few bacteria encode the genes to make that conjugation structure. But if you have the conjugation structure, then this will be a more dominant way that you exchange DNA. Transduction is another process. And this is where phages, so viruses that infect bacteria, are responsible for moving DNA and genes between two bacterial cells. And this happens because at a low frequency, when phages are packing their own genomes into their capsid, sometimes they can accidentally pack the host DNA into that capsid and then carry that into a new cell. But that's more of a mispackaging of bacterial DNA. Usually phages are just moving their own genomes from cell to cell. And then transformation is an additional way that bacteria can acquire genes horizontally. And this requires the bacteria to take up DNA from the environment. And so this is usually DNA that has been secreted, occasionally actively secreted by bacteria, but usually is in the environment because some other cell died and popped open. And so there's just the DNA present in the environment. And then bacteria have molecular machinery that can grab that DNA and take it into their cell. So summary of bacterial biology. There are two fairly major clades of bacteria that are gram-negative or gram-positive. And these have different cell envelopes, with the gram-negatives having the two membrane layer and the gram-positives having only one lipid membrane, but that really thick peptidoglycan layer. Bacteria are able to move through their environment if they have motile appendages like flagella or the type 4 pili, with the pili being the surface motility. And they also have appendages and extracellular polysaccharides that allow them to stick to surfaces and attach. Bacteria can display complex community behaviors even though they're single cellular. They have haploid genomes that are relatively small, 6 megabases. They have a high gene density without introns, and they're polycistronic, or they encode multiple genes on a single mRNA. And bacteria are largely asexual organisms that divide with binary fission, just cells dividing in half. But they can have genetic exchange through horizontal gene transfer. Okay, so based on what you guys know from other classes about eukaryotes, I'd like you to use the Zoom chat to highlight some differences between bacteria and what you know about fungi, plants, or animals, or eukaryotes broadly. And if you can highlight specifically fungi, plants, or animals, that'll be beneficial because those guys are all very, yeah, they're quite different organisms. Try to open up the chat. Yeah, so they have, I like that, types of cell walls. So animal cells don't have cell walls. Fungi and plants both have cell walls, but fungi, plants, and bacteria all have different chemistry to their cell walls. Eukaryotes have membrane-bound organelles. Yep, bacteria very, very rarely have membrane-bound organelles, but there's probably a weirdo that does. Yeah, so fungi's cell walls are chitin. Yes, the average size. So bacteria, I didn't highlight that, are about a micron, and eukaryotes are often 10 to 100 times that size. Yeah, so bacteria don't have DNA in their nucleus, but eukaryotes, the true, U means true, and karyo means nucleus, I guess. The Greeks knew that. I don't know, but eukaryote somehow translates to has a nucleus. Yeah, bacteria have the peptidoglycan cell walls. Yes, I like this one, different ribosomal structure. So we'll get to that in the lab lecture today. But the unique ribosomal structure of bacteria allows us to be able to specifically study bacteria when we do genome sequencing in environments, or DNA sequencing in environments. Ooh, that's a good one. Patricia says that she doesn't think anyone else has plasmids. And I like that there's a question mark, and I like that there's uncertainty, because it's good to be to acknowledge your uncertainty. There are other organisms with plasmids. I think we'll talk about some fungi that have some later, but there, yeah, so bacteria are the plasmid experts, but yeah, we tend to not highlight all the beautiful exceptions in biology. So we'll try to highlight some of those later this quarter. And then, yeah, cell compartmentalization. Bacteria have much fewer compartments. And I deleted my slides on this because it was too intense bacteriology. But I do want to say bacteria lack as many defined compartments, but they are still organized cells. People are starting to be able to do more focused microscopy and look at where proteins are in bacterial cells. And there is some structure and some cell biology organization in bacteria. So they're not just complete bags of enzymes. Okay, cool. Thanks for participating, guys. And I guess during that, I clicked to the next slide. So now we're going to move into talking about how cool and exciting plant-associated bacteria are. This is going to be general traits of plant-associated bacteria, although I will highlight a few genuses of bacteria. And I want to say that in this class, we're not going to ask you to be too specific about bacterial species level identification, but we do want you to be able to associate the genera. So like when I say Agrobacterium or when I say Xylella, know the bacteria at that level and where their traits are. And we're not going to be too picky about the species level because bacteriologists love to rename bacteria. And so some of the species level designations are still a moving target right now. Okay, so we're going to first focus on where bacteria colonize plants. We're going to talk about general strategies bacteria have for colonizing plants, and briefly touch on taxonomy and who these plant colonizing bacteria are. Although we'll go deeper into that on Thursday's lecture. Okay, so there's going to be a lot of location jargon in this lecture. Oops, I should not use the mouse because I can scroll on it. So epiphytic is a term that refers to on the surface. Epi is like similar to epidermis. Those are epidermal cells. They're the surface cells on the plant. Epiphytic is on the surface of the plant, with phytic being derived from a word for plant, Greek maybe. And so epiphytic bacteria will be ones that we find on surfaces. So that can be the surface of leaves, it can be the surface of roots, but it's on the surface. Those same bacteria, when they move inside the plant, will now be endophytic bacteria. And so endo is inside the plant. So these might colonize locations like the apoplast. We refer to the apoplast as the extracellular space between cells. All this space between these plant cells is the apoplast. Often people are talking about leaf apoplasts, but apoplasts can describe additional parts of plant anatomy as well. The endophytic bacteria also can colonize the vascular areas of plants. So the xylem, which is transporting water from roots to shoots, and the phloem, which transports a lot of the sugars from the leaves to around the plant. And endophytic bacteria can also be in the root cortex, or really anywhere inside the plant. And there are some bacteria that predominantly are on surfaces and others that are predominantly inside the plant. But these terms themselves are just describing the physical location of the bacteria. And it's also important to note that it's not making an assumption about whether it's a pathogen or not a pathogen. It's just a location. And so we only know if it's a pathogen depending on if there is a disease outcome. Okay, so bacteria can colonize many habitats on the plants, roots and rhizosphere, the leaves and stem, which we collectively call the phyllosphere at times. So phyllosphere being the above ground parts of the plant. They can colonize flowers, they can colonize the nectaries with the nectar inside the flower. Some can colonize the fruits and seeds of the plant. And some bacteria can colonize the vascular system and those endophytic compartments. And so xylem and phloem, as well as the apoplast. Okay, so why do bacteria like to live on plants? And at a simple level, plants are full of food. Just like most eukaryotic hosts are full of lots of tasty metabolites that bacteria would like to eat. So plants are able to undergo photosynthesis and turn carbon dioxide into sugar. Plants are able to accumulate a lot of mineral nutrients from the soil. And although they keep these nutrients confined from the bacteria, there's a lot of potential food there. And so that's provided a strong evolutionary pressure for bacteria to evolve mechanisms that allow them to get to that nutrient. But in addition to storing some of those nutrients, plants also are a little bit leaky. And some of this is potentially an adaptive strategy, although it's hard to, the microbiome community likes to talk about the plant as trying to recruit beneficial bacteria to certain organs, and maybe, but we have to be careful about those metaphors. So the rhizosphere is the region of the roots that's on the surface. And so rhizo stands for roots, and then sphere is, I guess, the, you know, the world of the around the roots. And this is a epiphytic location, and it's a really dense microbial environment because there is a lot of food that gets released into the rhizosphere. So estimates of carbon movement in plants indicate that plants can release up to 20% of all of the carbon that they fix into photosynthate. 20% of that carbon gets exuded through plant roots, and then provides kind of an oasis in the soil of a lot of organic acids, maybe some sugars that soil microbes can use to grow to high densities on. And so there's a lot of potential food here. On the leaf surface, plants are also releasing a lot of volatile organic compounds, volatile organic carbons, VOCs. So some of these are methanol. For plant beneficials, you'll find a lot of methylotrophs, bacteria that eat methanol. And some of those organic carbons being volatile, they're dry, like evaporating off without the plant being able to do much. So they can come directly through the stomata as the gas exchange is happening for photosynthesis. Okay, so we know that there's food available for bacteria and other microbes on plants, but how do bacteria actually invade plants? And this could be invasion by pathogens, but there's also some non-pathogens that will have these same strategies and similarly invade plants. The main ways that bacteria get into plants are through wounds in the plants, or through a handful of natural openings in plant anatomy. And it's important to note that bacteria are not as aggressive as fungi, nematodes, and insects. Bacteria don't have strategies for brute force entry. They're relying on wounds and natural openings, but these are fairly, abundant in a plant growing in the environment. Okay, so this is actually a wound on my plum tree because it was a really intense plum year this spring, and you can see like exactly how robust the plums were. We didn't really have our tree pruned that well, and the weight of the plums itself actually broke our tree. And I saw this around Davis a lot. A lot of plum trees went down this year because it was just too good of a year for plums, and they were too heavy. So this wound right here creates a great opening for all sorts of pathogens to get into the plant. And that can happen just by plant growth, especially if it's poorly structured. A lot of agricultural practices can introduce wounds. So in order to get our plum tree to grow the right way, we should prune it. And so our pruning shears can be a great way to introduce bacterial pathogens into the tree. So it's important to sanitize pruning shears between cuts in order to reduce the movement of potential pathogens around your orchard or even your tree. Some weather conditions can induce wounds. So wind and hail can both produce wounds. So wind can cause damage, like what we see here. And then hail, the damage of ice falling, miles down from the sky can really shred plants and induce a lot of wounds that then all of those epiphytic bacteria can get introduced to the endophytic compartments and move from being a commensal stage to causing disease. A lot of animal vectors can make wounds that introduce bacteria into plants. Some of these vectors, I forget the name of the beetle, darn. I think it's the cucumber beetle, Colorado beetle. There's a beetle that does is a herbivore of cucurbits, and so you can see it's chewing a piece of the leaf here, and you can see its frass, its feces on the leaf surface. And so this is an insect vector that can ingest this Erwinia trachyophila, this Erwinia wilt pathogen. It transmits this pathogen between cucumber plants. And it does it by the fecal oral route. So it eats the bacteria, and then the bacteria gets deposited in its feces, and then can migrate into these wounded areas, and then colonize the xylem and cause a wilting disease. There's also some insects that are sap feeders. So xylem feeding insects are able to move xylellus species from plant to plant. And then other insects that feed on phloem sap are able to move Liberibacter from plant to plant. And we'll be talking about Liberibacter in the case study next week. So you'll learn a lot more about that. Okay, so when I think about natural openings for plants, they aren't just complete openings. I like to think of them as gated openings. They aren't, these openings are not defenseless. Plants have a lot of ways to keep most bacteria from being able to enter. But there are some gates that clever pathogens know how to open. And so, and these gates can be locked, or maybe they're just, small openings in the more strong physical defenses that plants have. So for bacteria that come in through roots, they tend to not, go through any part of the roots. They tend to go through natural weakenings in plant anatomy. And some of these happen where lateral roots form. And so there's main roots, and then lateral roots are just side roots. So as all of these roots form in a fractal-like method, what happens on the cellular level is these stronger fortified cells end up opening up and giving way so that the additional new growth can form through. And so this ends up being places where bacterial pathogens can enter. Additionally, this is where nutrients will be leaking out from a little bit more. And so bacteria that are able to do chemotaxis can sense the slightly higher gradient of nutrients, and they can move along the root towards those weak spots. Stomata are also a natural gate that bacteria can enter through. So stomata are present on leaves, and they allow the plant to have gas exchange that allows photosynthesis to take place. So most of the leaf surface, most of this epidermis, is reinforced by a waxy polymer called cutin, and this cuticle, that waxiness of it prevents water and gas from moving through most of the leaf surface. But the plant can control how much gas exchange happens by opening and closing the stomata. And so this is a place where bacterial pathogens and non-pathogens that have an endophytic part of their life cycle, they're able to find these stomata and move into them. Another natural opening on leaves are called hydathodes. And the function of the physiological function of hydathodes for plants is that these act as release valves for the pressure of xylem sap. So if the plant has taken up too much water and risks having, exploding because it has too much water that's been taken up, the hydathodes will allow that excess water pressure to leave through these pores. And it's not a completely open anatomy, but it does allow water to get out. And there are some pathogens that have adapted to be able to use this as one of their main ways to enter the plants. Flowers can also be points of entry for bacteria. So plants have a lot of weak spots in their anatomy that allows them to put nutrients into the nectar, as well as help that seed form. And so that's a natural opening that adapted pathogens can take advantage of. And so we'll learn about some bacterial pathogens that cause issues with colonizing flowers. Okay, so I want you to think of these natural openings as gates. And then there's a lot of strategies that we have discovered that bacterial pathogens use to pick the locks of those gates. We can also call these molecular tools. They're basically different strategies that bacteria have to manipulate the plant biology so that they can get into the plant. One of the famous examples is this carnitine toxin, which Pseudomonas syringae and some other leaf colonizing bacteria use to open the stomata of leaves. And here I'm going to show you some microscopy. So this is what a stomata looks like if you do a leaf peel and visualize it on probably a light microscope. So we can see the stomata is open here. That's the pore, and that's going to be able to allow gas to come in and out, and bacteria can go into it. So this is not a completely, it's not an unguarded gate. The plant is able to sense some of the molecular pieces of bacteria. And when it does that, so when we add just a wild, a normal Pseudomonas bacteria, the first response from the plant is to close off that stomata and prevent entry by the bacteria. So it closes the gate. And so this is when the plant recognizes some, we can call them elicitors, things that elicit immunity. However, shortly after that, the Pseudomonas bacteria make this secondary metabolite, the carnitine toxin, and that actually triggers the stomata to reopen. And so that's like that's a direct strategy of the bacteria picking the lock to then enter the plant. There's also other strategies that bacteria use to get in through these weakenings in the plant. Most phytopathogenic bacteria secrete enzymes that are able to degrade those cell wall polymers of plants. I don't think anyone shouted out chatted out the plant polysaccharides, but we've got them here. So plant cell walls have a lot of pectins, a lot of cellulose, and a lot of hemicellulose. And a lot of work has gone into characterizing secreted enzymes that bacteria and also many fungi have that are cellulases, so those cleave cellulose, pectinases, which cleave pectin. There's also these cool expansin proteins that are not actually enzymes, but they're proteins that relax the hydrogen bonds between the cellulose. And plants have these as part of their natural development, that expansins are necessary to allow the cell wall to expand during growth. And a cool thing is that some bacterial pathogens have acquired these genes horizontally. And it's believed that those are doing a similar process to help loosen the plant cell wall to allow the bacteria to move around in the plant. And then something you'll run into especially I think as time goes on, in genomics era, sometimes we don't know exactly what we're looking at, whether we see a gene for a cellulase or a pectinase. But we can predict that it's something approximately in that category. And so people started to call them cazymes, which are carbohydrate active enzymes. And so one of the key things I want you to remember is that bacteria go through gates in the plants, but they don't drill through the fortified walls. And so plants have fortified polymers of lignin, suberin, and cutin. And the commonalities of all of these are that they're really hydrophobic and are not very easy to degrade. There are some fungi that can degrade them, but bacteria are a little bit lazy. Yeah, so there's also bacteria that colonize the starchy tubers of potatoes, carrots, any sort of starchy below ground organ. A lot of these cause soft rot disease, where you get this really nasty goopiness inside your inside the produce. And the way that these get completely goopy is that these bacteria are cell wall degrading experts, and they secrete a lot of pectinases that degrade the middle lamina pectin layer between all of these cells and just create this nasty goopy mess. And this is a convergently evolved lifestyle, the soft rot bacteria that's present in multiple bacterial genera, Pectobacterium, Dicea, so forth. There's also, there's other ways that bacteria move systemically in plants. So this right here are the xylem vessels. And the walls of the xylem are lignified. So bacteria can't move across the majority of the xylem wall. But there are these pit membranes that connect vessels to each other and connect the vessels to other parts of the plant. And xylem colonizing bacteria, xylem pathogens, secrete cellulases and pectinases that can degrade this pit membrane. And this microscopy shows Xylella fastidiosa cells moving through the pit membrane. So this is what the pit membrane looks like in a healthy plant. And then this is, you can actually see the bacterial cells in the very degraded pit membrane. And Xylella causes Pierce's disease of grapevine, which is a pretty big deal in California still. And then there's also some very cool ways that cell wall degrading enzymes have influenced evolution of bacteria. So the Xanthomonas genus, Xanthomonas are generally leaf pathogens, but they can cause different diseases depending on specifically which strategies, which plant pathogenic strategies and traits they're expressing. Some of the Xanthomonas strains cause leaf spot diseases where you get these localized lesions. And other Xanthomonas move systemically in the xylem and cause blights that actually move down along the venation of the leaves. And there's a very cool recent paper that found one trait that helps contribute to the different lifestyles of this pathogen. And this happens, this is an evolutionary trait that has evolved in multiple clades. So it's evolved between some barley pathogens, between some rice strains where there's some rice that cause the more local diseases, and other ones that are more systemic. And what they found is that the more systemic pathogen has one extra cellulase, and that helps contribute to its ability to move systemically. And when they delete that cellulase from multiple strains, you can see that there's much shorter symptoms. So here the bacteria are added at the edge, and they have an assay where they can measure how far down the bacteria cause disease over time. Yeah. Okay, so in this final section, I'm going to talk about some general strategies that bacteria have for surviving plant immunity. Because once they get in the plant, the other thing that influences whether they're successful and cause disease or just successful and grow without causing disease is whether they're able to circumvent plant immunity, because plants are not defenseless. And so there's two different directions that this interaction can go. We can go in the direction that the plant is able to recognize the bacteria and mount a response that kills it off. And this is what we'll see with pathogens in a non-host. They might get in, but then they're not successful. The other direction that, the other disease outcome is that the bacteria survive and cause disease. And so this is what happens when you have a pathogen that has the correct adaptations that allow it to be successful in that host, with as well permissive environmental conditions. But so the major strategies that bacteria have for tolerating plant immunity is first just a general tolerance, able to survive some of those stresses. There's also strategies that they have that allow them to evade. And then strategies that allow them to manipulate and control plant immunity. I crossed out brute force because that's not really a strategy that many bacterial pathogens use. Some things that help plants help plants kill off bacteria is plants are chemical experts, and they have a lot of antimicrobial chemicals that can keep non-adapted pathogens from growing. But bacteria that grow in plants have many mechanisms to tolerate these antimicrobial chemicals. Some bacteria have certain modifications of their cell envelope that decreases the amount that those chemicals can get into the cell. Sometimes the bacteria have enzyme pathways that can break down and degrade those plant chemicals. So they remove the antimicrobial chemical. And then the strategy that is very common as well is that bacteria have pumps that allow them to secrete the chemicals outside of their cell. And additionally, when bacteria grow in those biofilm communities, the polysaccharides resist the diffusion of these chemicals into where the cells are. And so, by slowing the diffusion of antimicrobial chemicals, that provides some tolerance. Some bacteria have an evasion strategy. So an example of this is Xylella fastidiosa. So this is a bacteria that stays within the xylem. And the xylem is actually a dead tissue and doesn't have living plant cells in it. And so by staying within the xylem, the model is that Xylella is able to cut down on the amount of plant recognition of itself and doesn't elicit a strong plant immunity. And so it can evade detection. But plant cells are able to recognize a lot of components of bacteria. So some of those components we learned about earlier, the flagellin, the lipopolysaccharide that's on the gram-negative bacteria. Plants have receptors that can recognize these components. We call these components microbial associated molecular patterns, or MAMPs. And they're just, components of a bacterial cell that plants can recognize. So plants have receptors that bind those components. And so one immune evasion strategy that bacteria have is that they have versions of flagellin and versions of LPS that don't bind to those plant receptors very well. And so that allows them to evade detection. And then for the manipulation strategy, there's some very really cool strategies that gram-negative bacteria do. So gram-negative bacteria are able to create this multi-protein structure that we call a type 3 secretion system. And they use that as kind of a hypodermic needle that they inject into the plant cell, into the cytoplasm. And then the bacteria takes what we call effector proteins, which have an effect, very clever name. So these effectors are then secreted through that hypodermic needle into the plant cytoplasm. And they can do many they generally change plant cell physiology in a way that benefits the bacteria. But a very large number of these effectors have been found to interact with different parts of the plant immune machinery in order to dampen immune responses. And so not all bacteria have a type 3 secretion system, but it's very common in Xanthomonas and Pseudomonas, Ralstonia, Erwinia, and many of the gram-negatives, but still not all gram-negatives. And we're not going to require you to know specifically who has the type 3 secretion system, but just know that if a bacteria has it, if you look at its genome, you can assume that it's manipulating the plant to benefit the bacteria. And then, but I don't want to leave it as, just, although I am a big fan of bacteria, most times the bacteria do not win. If the bacteria won all the time, we would not have plants. So plants have a lot of immune strategies that are successful and kill off the bacteria. And one of these we'll talk about later this year, this quarter. The hypersensitive response is a very effective way to kill off many bacterial pathogens. And this is when those plant immune receptors, the resistance proteins, are successful and bind to pathogens. And some of them bind to those same effector proteins that the bacteria is trying to use. And then the plants mount a very strong immune response that has a lot of reactive oxygen species, which are free radicals that can harm bacterial lipids. They kind of harm everything in biology. And so those reactive oxygen species and other antimicrobial chemicals can kill off the bacteria. And then the plant also kind of sacrifices part of its tissue and collapses it in hopes of keeping the bacteria from spreading further. So the plant can often be successful. And then I'm going to just briefly touch on some of the bacteria groups that we're going to talk about in the next lectures. And first, I want to highlight that for bacterial diversity, relatively few genera contain plant pathogens. So this is the current estimate of how many bacteria genera exist. It's almost 9,000. And of those, about 30 genera are associated with plant diseases. So overall, not very many. They're also distributed widely across bacterial diversity, although most of them are in the gram negatives and within the gram negatives within the proteobacteria, which are all these blue guys. And so this is not 30, but this is 14 genera that we're going to focus on in next lecture, where I want to highlight that these are all very, farther evolutionary groups. So here we've got family or order level classification, where I've just drawn cartoon triangles to represent that these genera are more closely related to each other than these other genera. And next lecture, I'm going to highlight how these different bacterial groups have convergently evolved to have overlapping disease traits and overlapping ecological strategies with transmission and some of their pathogenic strategies. But right here we can go ahead and see that most bacterial plant pathogens are gram negative, although there are some gram positives that have that thicker peptidoglycan layer. And we're not going to talk too much about the molecules, but they are like very fun weirdos. They actually lack a cell wall. They evolutionarily came from gram positives, so they lack a cell wall, and they also only have the single membrane. And we're not going to go into details on those, but the poinsettia disease that Dr. Dye talked about in the intro is one of these, one of the molecule diseases. Yeah, so gram negatives and gram positives. Okay, so if people have some energy before we go to a break, I would like you to try to speculate on either why or how plant pathogen, the trait of being a plant pathogen has evolved multiple times. And you can either talk about why, so like what are the selective pressures that would encourage this trait to evolve in multiple bacterial lineages, and then you can also